Hello, everybody, and welcome to the Frontiers in Science Deep Dive webinar, Adapting Crops for Climate Change. I am Laure Sonnier. I am the Executive Editor of Frontiers in Science. And the webinar today presents the Frontiers in Science article by Professor Shabala and Professor Palmgren on how using wild crops to regain the abiotic stress is key to achieving global food security and sustainable agriculture in the face of climate change. We'll hear both from the article authors and from our guest experts about the next steps for success. So it is my great pleasure to welcome all of you today. And I would like to extend a special thank you to Professor Shabala and to Professor Palmgren for their trust and for their leadership in bringing this very important project to life and to success. We are delighted to have given it a home in our flagship journal, Frontiers in Science. So, just in a few words, what is Frontiers in Science? It is our flagship multidisciplinary title at Frontiers. The journal focuses on transformational science key to accelerate solutions for healthy lives on a healthy planet. We commission exceptional peer-reviewed scientific articles from world-leading researchers. And then around each of these articles, we built a hub of multi-audience content, including viewpoints, editorials, policy articles, and also content for children and the general public. This is all designed to extend the reach and the impact of the journal across science and also across policy and society. No other publisher does this in the way we do, and we are delighted to be driving innovation in publishing so that critical transformational science can reach everyone in society and accelerate scientific progress for all. Today's discussion is going to be moderated by Greg Foot. Greg is a scientist with a first-class degree in natural science from Cambridge University. He's also an award-winning TV and radio science presenter and producer based in the UK. Greg, we are, as always, delighted to have you with us today. So over to you and enjoy the event, everyone.
Thank you so much, Laure. Hello, everybody. It's an absolute pleasure for me today to be here to moderate the deep dive. It's great to see so many of you in the chat already. We have close to 900 people registered to join us today from over 80 countries. So very, very welcome to all of you. As Laure mentioned, this deep dive focuses on the Frontiers in Science lead article entitled Adapting Crops for Climate Change, Regaining Lost Abiotic Stress Tolerance in Crops. It was published a couple of months ago in December 2024. The world's population is growing and we need more food to feed more mouths. However, the yield of the staple crops that we rely on is falling. One big reason for that is due to stresses caused by the climate crisis. Our crops are optimized for high yields rather than for stress tolerance. So sadly, they are ill-equipped to continue producing food in the face of increasing temperatures, droughts, floods. I mean, the list goes on. Yes, we could produce more food by following the model that we've used for decades, turning more and more land into agricultural land, or using more and more fertilizer. But this damages biodiversity and it threatens the planet's fragile ecological balance. In their lead article in Frontiers in Science, today's authors discuss our options for solving this problem by offering not one, but two strategies for precision breeding crops that have increased tolerance to these abiotic stresses, these stresses caused by non-biological agents like drought and flooding. As we'll hear, their two strategies are either to take domesticated high-yield crops and genetically alter them to cope better with the stresses, or two, to take wild plants that already cope well with stresses and genetically alter them to improve their yields. They argue that it's currently impossible to tell which of these two approaches will be most successful, so they recommend action in both areas. In today's session, we're going to explore these two opportunities to adapt crops for the climate crisis and to boost food security, and also discuss what is needed for success and the challenges the approaches will face. If you would like to dive even deeper into this research, please do check out the article hub. It includes an editorial by Professor Malcolm Hawkesford of the Rothamsted Research in the UK. He's going to join us later for the panel discussion. Also, a viewpoint article by Professor Francesco Loreto and Dr. Giulia Azzori of Italy's National Research Council. You can also, as Laure mentioned, find a lay explainer, infographics and a video, as well as an article in Frontiers for Young Minds, which is our journal for kids. I absolutely love that. If you haven't had a look, please do. To start off today's deep dive, we will begin with one of our lead authors, Professor Sergey Shabala, who will set the scene for today's discussion. He will then be followed by his co-author, Dr. Michael Palmgren, who will discuss the strategies for obtaining crops that tolerate abiotic stresses. Following these presentations, I will then welcome two guest panellists to join us to continue the discussion. The aforementioned Professor Malcolm Hawkesford, author of the editorial article, and Dr. Rosa Rivero of the Spanish National Research Council. Now let's turn our attention to our first speaker. Professor Sergey Shabala is Chair in Plant Physiology at the University of Western Australia, where he leads innovative interdisciplinary research in stress biology and membrane transport to develop resilient crops in the face of increasing environmental challenges. He has revolutionised the study of plant cell responses through the development of MIFE technology, a non-invasive technique for examining how plants cope with various stresses. And, as we're going to hear today, his research into plant stress physiology and adaptive mechanisms to deal with harsh environmental conditions such as drought and salinity, waterlogging, oxidative stress, nutritional disorders, it's increasingly vital as the effects of climate change intensify. Sergey's recent work has focused on the efficiency of phosphorus use in crops, studying a unique species of wild Tibetan barley that grows and thrives in the nutrient-deficient soils of the Chinese mountains. Sergey, thank you for bringing this article to Frontiers in Science. The floor is yours.
Thank you, Greg. My pleasure. And good evening, good afternoon, good morning, depending on the time zones. So I think I'll put the stage and I'll talk about the stresses and I'll talk about the importance of this problem. So in a very, very conservative estimates, abiotic stresses cost us over 180 billion US dollars in lost opportunities. And this is the number which comes only from 4 stresses, which are relatively easy to calculate. In reality, it's much more than that. And problem becoming even worse, because if we look at any model predicting the climate change, the severity and the frequency of the abiotic stresses will only going to increase. And the problem, as Greg said, that we were targeting for last few decades, if not century, the yield-related trials for optimal conditions. So we created crops which are responsive to fertilizers and irrigation, normal watering, optimal temperatures. We have plants which really germinate uniformly and produce a lot of the grain at the same time, but they are not really, that's what the nature likes, because it might just coincide with the major stress event. So we lost abiotic stress tolerance, which was present in wild relatives of crops, but later on we inadvertently lost it during domestication. So Mickey will be talking about these issues in details. I'll be talking about the physiological part of the problem. So let's take the drought as an example. In a graph here at the bottom, you can see that the amount of drought, severe drought events in 11 most populous countries affected by salinity drought is increasing in a very alarming rate. And if you look at these maps, depending on the climate model you want to employ, you can see that the area shaded in red becoming more and more and more over the map. So we are talking about the drought as most likely future scenario for most of populated countries, and by 2050, half of the Earth, which will host more than 4 billion people, will be under semi-permanent or permanent drought conditions. So we need to do something about that. And that means if we want to produce more food from these areas, and unfortunately that's coincide with the major producing areas, we need to rely on irrigation. Right now, irrigation really accounts for the 70% of the total fresh water used on the planet by using our agriculture, with a small fraction for animal feed. And if I take Australia, the country where I live in, and give you an example, we have 25 of a GDP in a country coming from only 0.4% of irrigated land. That means that 75% of GDP comes from the rest 99.6%. So obviously productivity, the difference in productivity between the irrigated and dry land is a couple of orders of magnitude. So in the next decade or decades, we will be not simply able to produce any food without irrigation. And in the last 100 years, the consumption of fresh water for irrigation increased sixfold, and we expect it even getting worse or stronger. So again, if you're interested in later on, you can look at some numbers. You can find them in previously published papers. But reality is that, yes, we increasingly relying on irrigation for the agricultural production. The problem is that when we do this, we inadvertently add some salt to the land. So fresh water is really a scarce resource and is hardly available in many places around the world. Right now, salinity affects more than 100 million of our 1,000 million hectares of agricultural land, and it's increasing with alarming rate. In fact, every time we use irrigation, we destroy between 2 and 3 hectares of arable land every minute. So my talk is 10-12 minutes. By the end of my talk, 40 hectares or 30 to 40 hectares of the land will be taken out of production by conventional crops. It's already cost agriculture $27 billion in lost opportunities. And it's really becoming worse and worse, as I said, because we need to rely on more irrigation. The problem here is that all our crops we have created are salt excluders. So when you do some simple maths and crunch your numbers, you can see that we're putting between 3 and 8 tons, or 6, 8, depending on the country, tons of salt per hectare per year every time. Unfortunately, we design our crops to take only 5% of that salt, and 95 stays in the soil. So one year you added 6 hectares or 6 tons, and you end up with 5.5 tons of salt sitting in the soil. Next year you add another 5.5, another 5.5, and 5-10 years from now, the soil become not productive and not suitable for modern crops, because that's how we design them. And that's not how nature deals with the problem. So why is that? Because, as I said, we targeted. It was our intention, we believed, I think falsely now, but the common view was that crops do not take sodium because it's toxic, and therefore we targeted some specific genes, particularly SOS1, which pumps sodium across a plasma membrane, or HKT, which takes sodium and unload it from the shoot and put it back to roots. So all the breeding programs, all the selection for the salt tolerance in rice, wheat, maize, barley was done by targeting sodium exclusion related genes. Well, we believed 20, 30 years ago that this is what we're supposed to do. But the problem is that it's really not working properly. To give you an example, and that's one of the very good example, very great paper by my colleagues from University of Adelaide. So they targeted the HKT15 gene, which is expressed in parenchyma tissue to remove sodium from shoot. And they observed beneficial effect, which was increase in the yield by 25%. The problem was that it was observed only in 1 of the 3 fields. In 2 was no effect. And even here, the reduction compared with non-saline controls was 50%. So the farmers, every farmer will say, no, this is not good enough. And this is because we targeted the one specific trait which we believed is important, but our belief is not necessarily what the nature does to deal with the problem. So when you look at the all modern crops here, we have example, a few of them, wheat and rice. You can see that with increasing salinity, 10 decisiemens is roughly 100 millimole, 20% of the seawater. The yield of wheat drops dramatically. Rice is even more sensitive. And this is dotted line where we expect the soil salinity will set at 2050. So wheat will be only 20% productive, maybe 30%. Rice will be impossible to be grown here. Some semi-domesticated species like quinoa will have really stable yield over all this regime of salinity, and Mickey will be talking about that later on. So what we did in our breeding is obviously wrong. And as I said, nature does it differently. When you look at the sodium content in the shoot in the halophytes, plants which are well adapted to highly saline lands, the sodium content in them is 3-4-fold higher than in sensitive crops in here. So salinity tolerance on this scale and sodium accumulation in this are inversely correlated. In fact, the most tolerant plants are able to handle more salt in their tissues, and that's not what we did for selection. So a problem comes now phenotyping, how we do the breeding, and that's really the misconcept which was done for quite a few years. Another thing, because we select varieties with low accumulation of sodium. Another problem is really that the phenotyping is done mostly and driven by convenience. So the breeders are after technologies or methods which are high throughput and productive. So they need to screen thousands or many thousands of individual organisms per day. Therefore, we have something which is easy and simple, and this is not necessarily has a predictive value. So this type of phenotyping we are talking here now with the whole plant imaging and image analysis is perfectly fine for agronomical traits such as a flowering time or leaf angle or stem architecture or whatever, but it does not really have prognostic value for stress tolerance screening. And this is hardly surprising because we know that there are many genes involved in control of a tolerance. There are hundreds of them, and they are expressed in highly tissue-specific and cell-specific manner. So my colleague from US, Hasedineni, published a paper 15 years ago showing very nicely the cross-sections, longitudinal and radial cross-section of the root, and the difference, and enormous difference in expression of specific genes. And yet we are mixing all together. So if I make some analogies, in the last couple of decades, we made enormous progress in evolution of transcriptomics, so omics methods. So we started with conventional when we just chopped the entire tissue and got something like the smoothie, and we have all the genes mixed. And later on, we moved to single cell omics when we can really separate different tissues and different cells in terms of the gene expression. And later, we now go to even further on spatial transcriptomics. And yet, when we do phenotyping, we do it by eyes. So in prehistoric times, we just picked up the plants which were taller and better and stronger looking. And later on, we start to do it with using microscopes and other things. Now we use drones or we use infrared cameras or different things. But nonetheless, it's still by eye. So we just replace our eye by the camera. So we do not account for cell-specific operation of genes. So that's a fine for validation, but it's really not appropriate, not suitable for prognostic value and to find out the genes which might be involved in the better performance of that plant. So I will finish with this slide, and I will advocate for cell-based phenotyping, and I believe this is really the answer, and I believe this is where the buck stopped right now. So we should forget about the convenience of high throughput screening, and we should really go for the methods for phenotyping which account for tissue-specific operation of particular gene. And that can be either using fluorescent dyes or any microelectrode single cell techniques. There are more than one technique which you can use. And to give you an example, this is a result of the quantifying the distribution of sodium, which is toxic ion, in different root zones of the barley plants. When you look at the mature zone, there is no surprises. Cytosol is largely sodium-free, 24 units here, and in a sensitive varieties, it's 75, 72. So 3 times more sodium here, and it's toxic. However, when you look at the meristem, you can see that the cytosolic content of sodium here is 4 times higher than in sensitive varieties. So essentially, if we just look at the whole plant sodium content, we'll miss these excellent varieties which are capable to send sodium, high concentration of the accumulate, send a signal to a shoot for adaptation, and trigger all the cascade of adaptive responses. We are missing it. We are missing it with whole plant phenotyping, so we'll never discover what genes are responsible for that. And this is really the problem where we're standing now. We are selecting for wrong traits, and we are really almost hitting a brick wall. So where from now? I think I'll pass the mic to Michael Palmgren, who will talk about the 2 possible options for the breeding.
Thank you so much, Sergey, for that overview of your lead article and for setting the scene for today's discussions. We are indeed going to turn to Michael now, your co-author and our next speaker. Let me do the introduction first. So Professor Michael Palmgren is Professor of Plant Physiology in the Department of Plant and Environmental Sciences at the University of Copenhagen, Denmark. Michael's research in plant biology and membrane transport research has significantly contributed to exploring the potential of wild plants for sustainable agriculture. He leads innovative research that explores how to accelerate the domestication of wild and underutilized plants to develop climate-resilient crops. And his work looks to assist global food security by harnessing the genetic diversity of wild plant species. Michael was knighted in Denmark for his contributions to science and currently serves as the Vice President of the European Academy's Science Advisory Council. His current research interests include the dynamics of photosynthesis under naturally fluctuating light intensities, new approaches for rapid phenotyping, and the long-term monitoring of plant functional traits. Michael, thank you very, very much for being here. The floor is yours.
Thank you very much, Greg. It's a pleasure being here today. We have essentially two strategies for obtaining crops that tolerate abiotic stresses. I mean, we have some very high-yielding crops, fantastic crops. They have been domesticated and bred for high yields and palatability during thousands of years. And we can try to improve those crops, yeah? So to make them more hardy, this is one strategy. And by bringing back lost genes that maybe they had in the ancient times, but they have been lost during selection, yeah? Or we can take crops that are already, or not crops yet, but plants that are tolerant. It could be relatives to our current high-yielding crops, and see if we can improve their yields. The thing about doing the other thing is that if we take plants that are already tolerant, we don't need to worry why they are tolerant and what genes they have to make them tolerant. But we can just accept that they are very hardy and try to improve their yields. So this is starting from scratch again. So if I can get the next slide. So this should show that during domestication, when we have selected for high yield and palatability, we have lost some genetic diversity. There has been selection bottlenecks. So according to the first strategy that was outlined, the question is how we can bring them back again. And this, what we have called the rewilding strategy, that is to see if we can bring some wild genes back into our highly domesticated plants to see if they can become more hardy. If I can get the next slide that starts with a lost salt. So you can have a lost salt tolerance gene. It can be brought back by crossing it into an elite cultivar. And there are many attempts to do this. So you find a gene that might have been lost, and you find it in a wild relative or a very early domesticated plant, and then you try by crossing and back crossing to bring it back again, and this can have some advantages. Yeah, of course, the first disadvantage is when you make such a cross is that you dilute the elite germplasm, but that can be brought back again, at least to some degree, by back crossing. This has been done with some success. Sergey already mentioned this example here with HKT1 from where there was some salt tolerance brought back. Yeah. And if I can have the next slide. So you can also identify lost salt tolerance genes, like in this example here from Jin Kang Su's lab, where they overexpress a lost gene that is associated with salt tolerance, and they get some salt tolerance. I mean, in this example here, it's not really salt tolerance. They improve salt tolerance. And such genes you can identify, for example, by GWAS, so genome-wide association studies, where you cross high-yielding crop, in this example here, tomato, and with a wild relative, and then you find genes that correlates, single nucleotide polymorphisms correlates with changes in salt tolerance. And when you have identified such a gene, like in this example here, you try to overexpress it. And you can get some salt tolerance by doing this, yeah? If I can get the next slide. And you constantly find new genes that are related to salt tolerance. Now, in this example here, using tomato, it focuses on salt tolerance, but the same is true for drought tolerance and other abiotic stresses. You constantly identify new genes that seem to be important for this process. And by trying to bring them back, either by overexpression or crossing or whatever, there has been some successes in getting more resilience. But if I can get the next slide, I mean, if you look at the whole picture, then, for example, for salt tolerance here, it's not that there is just one gene involved or one. The picture becomes more and more complicated. So such some biotic stress tolerance traits, they are encoded for by very few genes, yeah. But for abiotic stress, it seems that in many cases, for many of the abiotic stresses, a plethora of genes are involved, and they all have to be coordinated. And in this is from a review here on salt tolerance, where you see the number of transporters that are involved in controlling this salt tolerance. And this picture ignores also the regulation by kinases and transcription factors. Everything has to work coordinated to be coordinated to get salt tolerance. So a little, if I can get the next slide, so a little maybe a depressing view is that it seems to be no silver bullet gene for abiotic stress tolerance. So, I mean, the silver bullet gene, I mean, a miracle gene that when you overexpress it or introduce it, then you get high salt tolerance like in halophytes. Nature abounds with wild plants that tolerate the toughest abiotic stresses. I mean, worldwide, we have maybe domesticated about 200 plants for food consumption, but wild plants, a number about up to 400,000 species of wild plants exist, and they live in every biotope and every climate, every condition you can imagine. So plants have adapted to all niches present in this planet. So you can, so for example, one example here's just given some examples of plants that are quite hardy. So for example, Chenopodium quinoa tolerates extreme salt stress and extreme drought, and is very nutritious plant that is semi-domesticated, but has not really been undergone the same domestication efforts as our present crops. So we could benefit from such plants, yeah. And what would be required here would be to increase their yields and palatability, yeah. So if I can get the next slide, so it's very, when we looked into this and tried to understand how could we do this possibly, we noticed one interesting thing is that in the domestication process of the foods we eat now, it has taken maybe 10,000 years for some of them, yeah. But in that process, when we got these wonderful crops we have today, it's interesting to note that no new genes were added. It's a we got from wild cabbage, we got incredibly amount of vegetables, completely different looking vegetables, without adding any new genes. If you can get the next slide, it's also true for our cereals. We got our fantastic, highly yielding cereals now without adding any new genes. The old farmers just selected for genetic diversity and selected during repeated work, plants that were more high yielding and tasted better. Yeah. So if I can get the next slides about, we know a lot now by genome sequencing studies and from genome-wide association studies. We know a lot about what happened during this domestication process, and many of the effects that came was because some genes lost gene function. There was loss of gene function. So one, Greg, I see you now. Yeah, I'm running out of time. Okay, okay. We lost gene function. Yeah, this, for example, one important thing in domestication is seed dispersal. If plants lose their seeds, then you lose yields. So one mutations that lost where you lost seeds, that caused the plant not to lose its seeds were one factor. Yeah, and other mutations were in gene expression studies. So essentially, it's mutations. So we see that it's mutations or increased genetic diversity that has the basis of domestication. So then comes up the hypothesis: if we can identify genes that looks like domestication genes, so a domestication gene is a gene that, when mutated, gives a domestication phenotype. If we can identify a homologue of a domestication gene in a wild plant, if we mutate that gene, either to lose function or to alter its expression, maybe we will get a domestication phenotype, and that provides us with a key to get domesticated wild plants. Yeah. So some remaining questions we have here is, I mean, how many genes would we have if we go for this strategy? How many genes would we have to mutate? And what about traits that are controlled by many genes? What can we do there? And how much will are the low-hanging fruits that can bring us a long way? Yeah. And finally, is it realistic that we can achieve high yields in a reasonable time by domesticating a wild plant that can compare with those high-yielding crops we have now? Thank you very much for your attention. Yeah.
Thank you, Michael. Thank you very much to you. And of course, your co-lead author, Sergey, once again, for that introductory and those insights. Before I do start the Q&A, though, I need to welcome two more guests to the panel. The first is the author of the editorial article on the lead article, Professor Malcolm Hawkesford. Malcolm is the head of the Delivering Sustainable Wheat Strategic Programme at Rothamsted Research in the UK. He leads the Plant Nutrition Traits group there and the Rothamsted component of the Designing Future Wheat Strategic Programme, which is a cohesive national UK wheat research programme involving more than 25 groups of scientists at different institutions. Malcolm has been internationally recognised for his contributions in the fields of nitrogen and sulphur nutrition and transport, and is also an honorary professor in plant sciences at the University of Nottingham. Also joining the panel alongside Malcolm is Dr. Rosa Rivero. Rosa is a senior research scientist at the Spanish National Research Council's Department of Plant Nutrition. Rosa's research focuses on the regulation of plant cells under combined stress conditions in horticultural plants, employing a range of tools including molecular genetics and bioinformatics. Recently, she has explored the synchronization of plant metabolic pathways and the coordination of genes and metabolites under abiotic stress conditions, and has initiated research into the biochemical and molecular mechanisms by which melatonin induces plant stress tolerance. Welcome, Rosa, as well. Thank you for being part of the day. Let me start with a question to each of the two that I've just introduced. Let's go first to Malcolm. Malcolm, your editorial underscores the importance of strategies like rewilding and de novo domestication to bolster food security in the face of climate change. Reflecting on the findings of the lead article and what we've heard in today's presentations, how do you perceive the role of integrating stress-tolerant traits from wild relatives into staple crops, considering the potential scientific and policy challenges?
Okay, thanks very much, Greg, for that question. Really great question. Before I answer it, I would just like to say what a great article I think Michael and Sergey have put together, and I really recommend it to everybody. There's a lot of interesting concepts in there which address these issues. So, please look at it. It's clear that yield potential of most crops is actually rather good, and that that's the result of traditional breeding over the last 100 or more years. But the reality is that globally, almost everywhere, that yield potential is not achieved to a greater or lesser extent. Even in good environments, we don't really get the full potential, and in more stressed environments, it's well below that potential. And of course, climate change is just making things worse and worse. So we are trying to grow crops in suboptimal conditions. It's also true that most modern crops and varieties of crops have been bred from rather restricted germplasm bases, and they've also been bred under ideal conditions. So really going backwards and trying to exploit a wider germplasm base, particularly wild relatives or old ancient varieties, is really a great way to bring genes that have been lost in the modern pool, genes that could help grow crops in the stressed environments, particularly if we're looking at wild relatives that are already adapted to more stressful conditions. So I think technologies and techniques and approaches utilizing old germplasm is really the way forward. Wild relatives and two very good approaches have been proposed here. Certainly there are lots of difficulties. There are difficulties both scientific and also in terms of acceptance of technologies, which need to be overcome. And maybe we'll discuss some of these things over the next hour or so. Okay.
Thank you, Malcolm. Thanks very much. And now my question to you, Rosa. The lead article acknowledges the complexities of multigenic stress responses. So drawing on your research on the specific regulation of plant cells under combined stress conditions, how do you perceive the role of integrating comprehensive stress response pathways, such as those involving reactive oxygen species, osmoprotectants, metabolic synchronization, etc.? How do you integrate those into these broader adaptation strategies?
Thank you for the question. As you may know, and as a scientific point of view, I'm working with abiotic stress combination, and this abiotic stress combination is when you move plants from where the scientists used to work, like it's from control conditions, and then we apply an stress, and then we move to the field, and the things are not working as we were expected. So because if we can find in the field, one stress is not only acting by itself, it's just combining with other abiotic stresses. So in this context of the combined stress conditions, plants must employ an integrated regulatory mechanisms to ensure survival and optimal growth. So then integration of all the comprehensive stress response pathways, and as the Professor Shabala and also Professor Palmgren identify also and check in their talks, there is not only one gene or only a couple of genes working in this tolerance. There are many molecular markers working together, not only genes, also metabolites, also some proteins, etc. So in this sense, we need to take advantage of the knowledge that we are generating. And the thing that we observe in my research group is that when you combine two or more abiotic stresses, the plant responds in a very specific manner. So a pool of genes, protein, and metabolites just express specifically under certain conditions, and it depends of the abiotic stress that you are combining, but also the intensity and the duration of this stress. So it's very important that we take into consideration these specific molecular markers that are just expressed under certain conditions and sometimes climate regions, right? In this context, we have always observed that we have an overexpression of reactive oxygen species and accumulation of these species, also osmoprotectant, and some we observe also a really specific metabolic synchronization depends of the abiotic stress that are combined, right? For that, it is important to know what are the genes that are being specifically regulated under these climate zone conditions, but also what is the interplay that the main signaling molecules that are governing these tolerance mechanisms are reactive oxygen species, nitrogen, nitric oxide, hydrogen peroxide as a reacting oxygen species, and also the new molecule that we are actually discovering as a master regulator of the plant stress tolerance, melatonin, how all of them can work together and how can interplay and how they can govern these signaling mechanisms specific for abiotic stress combination. So I think that is very important that we focus in the next years our attention in how plants actually develop under field conditions and studying this specific molecular mechanism and how the plant react until this. And of course, we always have to go back to these wild relatives to get a very true insight of how these molecular mechanisms can act in field.
Thank you, Rosa. As you say, lots going on in field conditions, lots of factors being combined that you need to kind of try to unpick. And let's try to unpick some of this conversation. So I need to put some of your questions, our live audience's questions, to these fantastic panelists. I'm going to suggest that we start wide and we focus in. So I'm going to take questions first on the impact of climate change on crop yields and food security, big picture. Then we'll discuss further the strategies that has been introduced, rewilding versus de novo domestication. Then let's move on to the complexity, kind of what Rose was saying there about the complexity of the stress tolerance and the genetic role in that. Then we'll focus down into cell-based phenotyping. And then at the end, we'll come right back to a discussion about public perception and what we can do about that. That is the plan. Okay, let me put this question first, and I'll put it to our authors first. So someone asks, how can we quantify and address the impacts of various climate-related stresses such as heat, drought, salinity on staple crops, and what are the most promising short-term strategies for farmers to mitigate these effects? Sergey.
Right. I think there are two questions here. First one is quantification. So for some stresses, it's not really easy to do. For example, heat stress, you cannot really there's nothing to have as a control, right? So it's a background temperature. But normally you combine or you compare stressed and unstressed conditions. So what Malcolm said was a yield potential in every crop, which really what they can produce under what we believe optimal conditions. And they never reach it in under field conditions. So essentially we can calculate how much we might expect from 1 hectare and what we are getting there and convert it into monetary values. For heat stress, we know how much yield is lost for 1 degree centigrade increase in ambient air temperature, for example, during the night. And we can extrapolate it to climate models and say in 15 years or in 20 years, we expect 27% yield loss in soybeans due to the increased or elevated night temperatures. And we can say, okay, that will cost that much farmers based on, commodity price. So this is a way how we should do it. It's really difficult to find in literature. I published a paper a couple of years ago, 3 years ago, I think, 2021. And I spent like a month just browsing through different journals and things, trying to get this economic information, because plant scientists normally are not good with that. And economists are really looking from different angles. When in terms of mitigation, there are two simple answers. It's agronomy versus breeding. And it's really a combination of that. The problem is that many farmers are very conservative. They want to use the same varieties their parents and grandparents are using. There are IP issues around these transnational, which really produce hybrids, and you need to buy seeds and they don't want it. And the agronomical measures are limited. So I put my money into breeding, but it's not the immediate answer. So it's a combination of both approaches. And in third world countries, people simply prime seeds to get them more salt tolerant, and that works to some extent. Or you put the lime in the field and you reduce the impact of salinity stress. Of course, you use irrigation to mitigate against that stress, but there's a limitation on that. And what we are trying to argue in the paper, that at some stage we'll come to the point where the mitigation by agronomical means will become not feasible, and we hit a brick wall with trying to use the diversity, available genetic diversity of existing crops. So we need either to do changes in legislation and allow these GM-like technologies to be, or need to force public to accept them as the only way to go, or we need really a small miracle, a silver bullet, which Michael was talking about that, which is unlikely.
Well, we'll return to that conversation at the end of the panel. Thanks, Sergey. Let me put Ali Baghdadi's question, perhaps to you, Michael. What are the current and projected impacts of climate change, including biotic and abiotic stresses, on cropping systems, and what are their implications for the future of agricultural productivity? Do we have any facts and figures or any sense of this? Michael, is that something you can answer for us?
I think that's more a question for Sergey. He has looked into this, yeah. So sorry.
The king of the stats. Yeah, no worries. Sergey, give us a couple of quick bullet points on that.
Well, I think from the top of my head, flooding stress or waterlogging stress, I mean, there are differences between how much submergency is in there, cost the world about 60 billion euros per year. Drought is 80 billions. Salinity is close to 30. So as I said, there are three stresses, these plus ozone, getting in excess of 180 billion per year. It's significant impact. It's huge impact. If you add here frost, cold, heat, diseases, we are talking about very significant amounts. We talk about huge amounts. And the problem is that we don't see it because most of the science, again, is probably, I'll probably keep these comments for the last session about social, because I'm going to be critical about the governments and funding bodies. So I think I'll skip that.
That's a good tease for that later chat. Keeps everyone on tenterhooks. Rosa, a question here I think could be for you. So Miles Curl says, what, in your opinion, are the main traits to be phenotyping when we're exploring these abiotic stresses, especially drought, tolerance, etc., in major crops such as wheat? Wheat, I'll come back to Malcolm on.
Well, thank you. We don't have just a trait to phenotype. We had several physiological cues that plants gave us when they don't feel comfortable in an environment, right? But there is a beautiful paper that has been published by Ron Midler very recently, I think last month, that it talks about how stomatal conductance is very different from the leaves than from the different part of the flower. And this is an important issue because flower is the end of the product that we are going to have the fruit, right? So if temperature, for example, heat stress is being differently controlled in leaves than in flowers. So probably we need to change our concept of how to measure some physiological parameters when we want to check for stress tolerance and go exactly for the plant part that we want. Actually this final product, like the fruits, right? And how it can be controlled. So if the physiological parameters are different in different plant organs and are being regulated in a different manner, probably the gene set and the metabolite set is also different. So probably can be one new way to explore there, no? to look for new traits of stress tolerance in plant in the field. And also when we are talking about abiotic stress combination, which is different, depends on the abiotic that is being combined, the intensity, the duration. I mean, every single point is, or every single state that you can measure can be different. So we need more research here. We need more people involved in the characterization of this abiotic stress combination and the specificity of this abiotic stress combination in the plants, and in order to get more cues and more insight in this and the specificity of the molecular mechanisms and signaling mechanisms that is involved in the tolerance.
Again, a complex situation. And Malcolm, with your kind of experience, your expertise in wheat, because that was something that Miles asked about at the end of the question, do we have any more knowledge, or is it just as complicated with wheat? You know, any main traits we want to be phenotyping?
Yeah, there are key traits, and a really important approach that a number of my colleagues take is combining both crop modeling and climate modeling. And when you do this properly, you can actually then predict which components are going to be susceptible to different climatic conditions. And you can do this in a region-specific manner, which is really important because different parts of the world are going to suffer different stresses. One of the questions that came from the audience was, well, how do you cope with all these different kinds of stresses? You know, you might be talking about drought, and in some parts of the year, or in some years, drought may be an issue. Other years, and even in the same locality, you may have flooding issues. So it's quite difficult to predict when you get these extremes over short terms of period, but long term you can predict in different regions the impacts of climate change and what they're going to have and what stresses are going to be apparent at which stages of development. Because you can have heat stresses and you can have heat events which cause problems in fertility or in grain filling, and these can be quite short events but can have major impacts. And modeling can help tell you if heat stresses are going to be more or less common at those particular developmental stages in whatever crop. I mean, we work particularly in wheat and we can make predictions in that way. So you can identify which stresses may be the most problematic, but you can never pin down all of the stresses. And then just one other thing that just came to my mind is in the short term, I do think agronomy is important, and that farmers being educated in ways to cope with some of these different abiotic stresses has got to help in the short term, because most of the genetic approaches that we're talking about are actually rather longer term solutions.
Thank you, Malcolm. Again, we'll pick that up at the end, but that's such a good point that stresses change, the combination changes. Some are short term, but they have a bigger impact that some, so again, complexity. Let's move on to the strategies. Let's move on to part 2 of the conversation, the strategies for adapting crops. We've heard about rewilding versus de novo domestication. So let's dive into that a little bit. Michael, here's a question for you from Sebastian, which says, what role do wild relatives have in transferring traits to the cultivated plants? And how do we explore these wild relatives in the natural world?
You hear me? I'm muted.
Yep.
Okay, that's a very good question, and I mean, I don't know how to answer exactly, but I mean, if you an example, now we talked about salt tolerance, but if you take drought tolerance, so for example, barley, Hordeum vulgare, it comes from Hordeum spontaneum. Yeah, it's the wild relative of Hordeum vulgare. And in the Negev desert in Israel, grows ecotypes of Hordeum spontaneum that grow happily in the Negev, in the desert. Yeah, why they are so drought tolerant, we don't know. We don't have a clue. Yeah, maybe there's some ideas, maybe hypotheses, but it is drought tolerant. Maybe not salt tolerant, maybe not flood tolerant, but here we have a relative to barley that is highly drought tolerant. And we know a lot about the genetic changes that made barley a highly yielding crop. Yeah. So again, the hypothesis would be that if we could introduce mutations into those ecotypes of Hordeum spontaneum that grows in the Negev desert, we would likely get a more, at least more yielding. We should theoretically be able to turn it into a crop. Yeah?
Thanks, Michael.
I don't know if that was an answer to the question.
Yeah, it helps us explore it. Let's go, Malcolm. Let me put this one to you from Ches de la Cruz. The question is, what are the latest advancements in mechanization and automation efforts in breeding operations, and how are they enhancing efficiency and effectiveness?
Okay, well, this is something that Sergey won't like, but whole plant phenotyping is still the way forward as far as most breeders are concerned, and even the mechanization of that is difficult. It's very often expensive and requires a lot of expertise, and so mostly there is still a lot of manual breeding by eye and picking the best varieties. And of course, the key targets in commercial breeding are still intrinsic yield. So everybody's after yield, and we've already heard about what the problems are there. And of course, also pathogen resistance, which is another kind of stress, which also interacts with changes in climate. So I think you asked about the mechanization. I mean, there are some nice technologies around. You know, we utilize some ourselves here at Rothamsted, and there are a lot of new tools about, you know, drone-based tools, for example. But it still comes down to needing to look at individual plants, and if Sergey and Michael to be believed, looking down at an even higher resolution. And it is a key about resolution, but you have to offset resolution and throughput for the most part. And those are two quite difficult things to resolve. Sort of half answered your question, I think.
Well, you also set up a response for Sergey there by starting by saying it's not going to like this. So yeah, whole plant phenotyping. Go and Sergey, over to you. Thoughts, reactions?
No, I think, look, what we're talking about different types of phenotyping. So I would probably call what Malcolm and Rosa said before, approach like horses for courses, right? So we cannot make the universal plant, which will be extremely tolerant to every stress and every stress combination. Well, will be such plant which will be surviving and not growing because there will be no energy left for anything else except the defense. So what we need to do, we need to accept some reasonable yield penalties under conditions which are rare. Let's say you have one of 100 in 100 years event. Well, bad luck, right? It's like you smash your car, you don't do it every day. So that does not prevent you driving to and from work every day, right? So if you want to do the breeding, you need to predict, okay, this region, this part of the world is prone to periodic flooding and heat waves coming in that particular month. So you can play with phenology, you can play with other things to avoid the stress because it's not tolerance, it's avoidance. So what Malcolm was talking about for the whole plant phenotyping, this is exactly what the point is. By looking at the plants which are tall and mature or flower at particular time point, you can shift the flowering time away from the drought or heat stress and survive and produce normal yield. So whole plant phenotyping is perfectly suitable for these purposes. However, if you want to deal with tolerance, and that what will be needed 10, 20 years from now when avoidance become impossible, you need to move from plants which are tall and greener to those which we can quantify operation of specific genes. So essentially here it's like a puzzle. You need to put together 5, 6, 10 key genes or core genes which are responsible. And Rosa mentioned Ron Mittler and ROSs. Yes, it's a master switch there. NADPH oxidase and RS production is upstream of every signaling cascade. Pathogens, salinity, drought, chilling, flooding, doesn't really matter. ROS, hormonal regulation, calcium signaling, they all interact and they're there. If we will find a technology which will allow us to screen for efficiency of CIPKs or CBLs, right, in regulation of these transporters, we'll be able to find the QTLs or genes or whatever responsible for that, or QTLs, and do it via conventional breeding. So there's no really, it's not mutually exclusive, it's complementary, two complementary approaches, and that's what we need to target. But again, in a search for high throughput, the breeders, I've spoken with quite a few of barley breeders, no, they ask me, how many plants per day can you do it? And if it's less than 500 at least, they're not willing to talk. And my usual answer to them is, give me money and I'll do a robotic system which will have 100 channels in parallel and I'll screen you 1,000 per day. It's all come to resources. We simply do not allocate them because we still like 20 years behind in phenotyping compared with the omics and genotyping. So that's my main point. We are behind in one technique while we have, so we built Ferrari with a nice, powerful motor, nice shiny wheels and everything, and we forgot to put the petrol in the tank. So we know Ferrari is not running because we don't really have the missing component.
Thank you, Sergey. I'm enjoying all the horse-related comments, not only horses for courses, but Ferrari, of course, which has a horse on it. Right. There are some really good points about genes and genetics there that I think we can come back to. If you're listening to this and you'd like to add your question, please do add it on the Q&A tab there. I've got one more kind of broad question before we focus down. This was from Francesca Dusclini, who says, thank you very much to all the speakers. Very interesting talks on the new trends in plant breeding and improvement. I have a more general final question. Given all this data, how would you define resilience? And what aspects of it should we focus on, since they are all connected and depend on one another? Rosa, what do you say to that? How would you define resilience if they're all interconnected in the way that you've already described?
Resilience is a word that is appearing everywhere right now. I mean, the last 2-3 years, before it was resistance and tolerance, and now resilience. So resilience is, I think, the capacity of it can be a plant, a person, an animal, whatever, to adapt to something or to some environment or to some situation, right? That is, and for that, I mean, as a person, you can move and you can find another different environment, and you go to the heat outside to your office, which is cool and nice. Or as a plant, you just can, synthesize a lot of compounds, activate a lot of genes that are able to synthesize this compound that is gonna try to avoid you about the osmotic stress that is being generated by salinity, by heat, by drought, but many different things, and act in consequences. So for me, this is resilience, and you get the resilience when you are able not to fully recover your capacity of growth, not growth 100% as if you are, in a perfect climate condition, but resume your growth that you stop when you feel the stress, and then continue growing and producing at least some flowers and some fruit or seeds, right? Just to keep your generation going and going in the next year. So this is for me resilience, and how we can get to this. We have been discussing this the whole morning, many different ways to do it. We need to check. We need to go to the wild relatives. We need to do the breeding techniques and modernize us in breeding techniques using all the molecular tools that we have available, and try to combine all this knowledge in order to keep going. But like Sergey said before, phenotyping, which is one of the main key of find or define resilience and tolerance in plants, is being really back in. I mean, it's not developing as faster as the other technologies. And we know a lot of genes. We know about the proteins and how the proteins interact and interplay one to each other, but we just recently know that, for example, like Ron Miller said, that leaf stomatal conductance is going to be different from the petal and from the sepal of a flower. So,
well, let me jump then, Rosa, into that question about phenotyping that you've raised for us, and what you mentioned about complexity. Michael's very kindly put your hand up, and I have a follow-up question in addition to what I think you're going to say, Michael, which I'm going to throw to you so you can answer both at the same time. We've got a question from Michelle Hernord, who says, we've been mainly focusing on managing single stress factors affecting crops. Let's come back to the multiple concurrent stresses, and to bring it to phenotyping, how can we effectively approach phenotyping and breeding in these scenarios? The floor is yours, Michael.
It's just related to resilience and multi-resilience. There's a very good comment from Christian Thorup Christensen here in the chat that while plants can be surviving under stress does not necessarily show that they can also be productive crops. I think this is a very relevant comment, because I think we have to also keep in mind, before we get too optimistic, that there's a trade-off with everything. I mean, there might be a cost of resilience. So maybe if we introduce resilience in a crop by either one strategy or another, maybe we cannot expect the yields to be as high as those crops that have been bred for yield only. Yeah. That's maybe one reason why they have lost their tolerance genes, because they were associated with lower yields. Yeah, because there is a trade-off. There's a cost of everything. Yeah. So in a sustainable agriculture where we have crops that tolerate maybe more that are more resilient, maybe we have to accept the fact that they will maybe never be as productive as the present crops.
Yeah, good point. Malcolm's got his hand up now to continue the chat. Go on, Malcolm.
Yeah. Just to, I mean, my definition of resilience, and one that would be sort of relevant to farmers, in fact to everyone, would be productivity. This also comes back to Christian's comment, productivity under different conditions. So under different year to year, so a yield stability. So even under stressful conditions or in stressful situations. So that's my idea of resilience. And then just to also pick up on some of the questions in the chat. For phenotyping, someone asked, well, what about machine learning, artificial intelligence? These are really important approaches going forward and which are currently utilized. So most come back to whole plant or whole plot. Phenotyping these days is done using imaging technologies with different sort of spectral specificities maybe, but nevertheless imaging technologies. And in terms of high throughput screening, without a doubt, machine learning technologies are a way that we can extract the information and extract ultimately the breeding lines in a breeding program from this high throughput phenotyping, such as image-based stuff.
Thank you, Malcolm. You may have frozen for me. I can still hear you though. Sergey, you've got your hand up. Over to you.
Yeah, I wanted to add to the comment about the combined stresses phenotyping for them. So if we just use one example, for example, if we want to screen or phenotype plants for their combined heat and drought and salt stress, one of the common thing in here will be the operation of the stomata and development of stomata. So to deal with this stress, plants need either to get more water to cool themselves down, and therefore they don't really suffer from water shortage, which is tricky. So if the stress should be supposed to be short, just 2 days, heat wave, they can rely on this resilience. If not, they need to survive, so they shut down stomata. When stomata close down, there is the residual transpiration still. Some water still evaporates through the surface, even then all the stomata closed. So the fewer stomata you have on the leaf surface, the more unproductive loss of water you have. And that's what is currently being targeted by few labs around the world. Julia Gray in UK is one of them, for example, to breed more drought and salt resilient wheat and rice, for example. So the development of stomata, which is controlled by probably a dozen of developmental genes or even less than that, is clearly a possibility to deal with at least 3 stresses, each of which has water component. So similar examples can be given for other things. And obviously it's possible because we're not talking about something unique. Halophytes or xerophytes, naturally drought-tolerant plants, they have these packages or combination of 5 or 6 most key traits. All we need to do is to screen for their operation in crops. And that's what we come to phenotyping. So once again, we need to look at what we believe is most important for particular environment and use a suite of phenotyping approaches, which will be specifically related to operation of these traits or these genes.
Thank you, Sergey. I don't know if anyone spotted the clock, but we've now only got 15 minutes left. I told you the time would go quick, so I'm going to do some quick fire questions, quick answers if I can. You've just introduced phenotyping, Sergey, so let's jump into that a little bit. And there's a really good question here that was actually sent in by email. We're taking them from all places today. What specific tools and technologies are utilised in cell-based phenotyping, and how can they lead to more precise crop breeding programs that enhance stress resilience? So we've touched on some of these already, but I'd love just a couple of bullet points from each of you. Malcolm, let's start with you.
Okay, I'm the worst person to ask because I don't do cell-based stuff, I'm very much a person who looks at performance in the field of plants under different conditions using image-based technology. And I think even if you increase the resolution of image-based technology, you can get down to even higher and smaller and smaller components of a plant. I can't really say any more than that.
No problem. We'll get that context now. We'll move into the specific tools and technologies then. So, Michael, what would you list as a couple of the specific tools and technologies for cell-based phenotyping?
I think this is more Sergey's domain. Yeah, cell-based phenotyping is to identify components that contribute to stress tolerance. Yeah, and and that is to find the remaining pieces in the puzzle that we maybe need to add to high-yielding crops to make them more productive. But the de novo domestication strategy is not based on such knowledge. The strength of this, if it works, is that you don't need to know anything about why Spontaneum thrives in the Negev desert. You only need to know how you only need to focus on increasing its yield. Yeah. So these are completely different approaches. Do you need to know about the genes that are responsible for stress resilience? This is required in order to make our present crops more productive. But if we base our research on those plants that are already resilient, no matter what mechanism, then we don't need this information. So it's completely different strategies. Yeah.
Let me just get a quick response from Malcolm on that one. Have you got your hand up?
It's because I forgot something. So I think a really interesting cell-based technology is being able to do single cell transcriptomics and spatial transcriptomics, because then you can really focus in on individual cells and tissues and how they're responding to different stresses.
Thanks for that addition. Go on then, Sergey. You are unmuted and ready to roll on the phenotyping question.
Yeah, I think the answer is in my 500 plus papers published, but in very brief, like we use in my lab two basic approaches for this cell-based phenotyping. One is using fluorescent dyes. So for example, again, I'll use salinity as more clear example. So the key for salt tolerance, true tolerance, not exclusion, is putting salt load in vacuole. And there is a specific gene, NHX transporter, which pumps sodium into vacuole. Huge amounts. It can be up to half a mole in there, in halophytes, even 1 mole. And we can quantify it by loading the fluorescent dye and have scanning the fluorescent microscopy with high resolution to see how much in a cytosol, how much in vacuole. So you can really make like robotic arm, you can put specimens one after one, get the fluorescent image, quantify the signals automatically. In fact, I'm putting together a proposal to the Research Council with one of the company in Czech Republic for that exactly approach. So it will be high throughput, well, moderately high, but still higher than manual. And another one is basic technique. You mentioned MIFE when you introduced me for microelectrode ion flux estimation. So essentially we have the thin capillary, thinner than your hair on your head, filled with ion selective cocktail with tip diameter 2 or 3 microns. And we can position next to a cell and we can count number of ions crossing there. So if we believe that's really pumping sodium out, we can tell which cell pumps sodium more and later do what Malcolm just said. Look at the spatial transcriptomics and look at the specific gene patterns in that cell compared with ours, find the difference and use it in our breeding programs. So techniques are in place. It's just the lack of high throughput, which really make them not commercially used, if I talk this way.
Let me ask you a follow-up that Maurice has just posed. Considering the vast diversity of issues, cell types, ions, structural features, as well as the need to screen large populations of plants, cell-based phenotyping appears to be low throughput. How can cell-based phenotyping be effectively implemented in practice, given these potential bottlenecks?
Should I answer that?
Yeah, please.
Yeah. Okay, the Nobel Prize for was given in 1993, Nobel Prize in Medicine, to Erwin Ney and Brent Sackman for the invention of the patch clamp technique. So it was first introduced in plant physiology in 96, and it's still manual work, and you can do 5-10 cells per day. Pharmaceutical companies took it on board, and now they have their automated system called Barracuda, for example, which costs $3 million and can do 396 channels per at the same time. And the operating cost is $12,000 per day. So throw some money in our direction and we'll make automation. That's as simple as that.
Brilliant pitch. Brilliant pitch. Right, I have less than 10 minutes, so let's move on to the public perception discussion, because we've touched on this already. You know, the successful implementation of new crop technologies hinges on public acceptance and understanding. And Rita poses a question. I'm going to turn to you first, Malcolm. What strategies could make genetically edited crops acceptable for commercial cultivation?
Okay, so I think some of the newer technologies for genome editing, have a lot less sort of baggage with them, so are going to be much more acceptable. I think the most critical thing are the traits that we tackle. They need to be traits which the public are on board with. So green technologies, fantastic, Food security, one would hope, is pretty high on the agenda for the general public. If it's just about making money, for big corporations, then that's really going to be problematic. But I think it's absolutely critical that we all, and I think most of us do, who work in any kind of related area, talk to the public, talk to the media, talk to children, and share our enthusiasm and share our knowledge, to explain to people that these technologies are nothing to be frightened of, and are something to be on board with. And we have to take the general public and the politicians with us. Otherwise we're not going to be able to implement any of these strategies, to be honest.
Brilliantly put. Thank you. And the other guests on the panel, what are your thoughts on this? What are the current challenges that are preventing either the research being done or the research being actioned into the wider world? Who wants to comment on that? Michael, Rosa, Sergey, anything you'd like to add to this? Sergey's unmuted. Off you go.
Yes,
Yeah, please.
I can make one comment. I think the problem is that most of the funding for science and research in this direction comes from first world countries with rich of resources, and we don't have food supply as a problem here. So people do not realize, they just believe that the food appears on the shelves in supermarkets by itself, essentially. So we don't really have the food shortage. COVID was an exception when the shelves are empty at some stage, and still it was not production, it was a delivery problem, right? So we have enough. The problem is that majority of the world lives in countries where climate is not really suitable for agricultural production, and the supply chains are not very often very efficient. So unless the governments and people in rich countries realize that they should be the world citizens rather than just look at their own particularly part of the world and be happy. I mean, ecology is important, the environment is paramount, but human lives are probably higher than that, and we have people dying from hunger because of complexity of logistics of getting the food delivered to these poor kids in Africa, for example, in sub-Saharan Africa. So I think we need really to step over our ambitions and principles, or ideology. Oh, we not accept what not natural. Like I'm not advocating back to GM crops, but some of the technologies are just classification is illegal. Like these, CRISPR editing in some countries it's non-GM, and in some it becomes a GM technology. So there's uncertainty among my colleagues in China, for example. They believe, oh, we really should not go this way because in a few years it might become, classified as GM, and therefore we have really wasted our time. I think it's more like social issues rather than scientific issues, which really are major hurdles at the moment. That's my view.
Thank you. Thanks for that. And just quickly, let me squeeze one more question in from Jenny, because it links to what you were saying. Jenny says, thank you for the presentations. Which approach would be most feasible for researchers in low to middle income countries? Who would like to take that as a quick answer? Anyone like to comment on that one?
Maybe
Michael?
I can.
Yes, please.
I mean, there's already genetic diversity present that you can select for, and you can increase genetic diversity by mutagenesis. It doesn't need to be directed. By molecular methods, you can look for a specific mutations that are present in the population. Yeah. So the improving yields of already tolerant plants does not require transgenes. So this is at least something here. And you don't need, in principle, you don't need to use CRISPR and new breeding techniques to introduce specific mutations. If you can increase genetic diversity and select from the genetic diversity, this can be done by molecular methods.
Thank you, Michael. Sadly, I think we're almost out of time, but I do have one last question. One last thing I'd like to throw at all of you. Let's wrap up. And to wrap up, I'd like to ask each of you for one final takeaway message, something that you'd like to either say to the scientific community, there's a lot watching, to a relevant agency or to a policymaker, or indeed to a member of the public. what is the one thing you would like to say? Let's go in reverse order of the way you appeared before. So I believe that goes Rosa first.
Okay, so for me, I mean, we need to integrate all the molecular tools that we have, all the phenotyping tools that we have. We need to go, all the scientists, we need to get together and, you know, and put together all the tools that we have in order to find, you know, common tools in different world climate environments. And like Sergi said before, and I want to just to add this, we don't have, I mean, the developed countries, we don't have the problem food production, not either in the supermarket, in our crops on our farms. We have to face also the problem of food waste most of the time, because we are not able to process all the food that we produce. We need to go to those countries and those regions that actually are facing true climate change, and they have land desertification and true problems with the climate. So that's my opinion. We need to integrate, and we need to go, all the scientists that from the different disciplines go together in order to find a common solution.
Thank you, Rosa. Malcolm, what's your one final takeaway message?
Okay, my final takeaway message is that resilience or stress resistance, or whatever we want to call it, you know, maintaining productivity under difficult conditions, there are not going to be easy solutions. They're all complex traits, and we have to really embrace the fact they're complex traits and require, you know, a lot of research. And I think it was, I hope I got the right person here. I think it was Sergey who said, although it might have been Michael, there are no magic silver bullets. And that is absolutely the case. You know, these are complex processes that are going to require fairly complex solutions.
Thank you, Malcolm. And Michael, what is your kind of final thought? What do you want to leave people with?
Yes, I think, I mean, for a future sustainable, resilient agriculture, I think we need to increase crop diversity. And I think in nature, you have plenty of solutions to many of the biological solutions to how to increase resilience. But I also think we have to accept that if we domesticate new plants or introduce tolerance traits to already existing crops, we will maybe have a trade-off. So we can maybe never expect to have the same yields as those crops that have only been selected for yield.
Thank you, Michael. And last but not least, we started with you, Sergey. We'll finish with you as well. What's your final kind of takeaway message to the scientific community or the agency or the policy or the public?
Yeah, well, I fully support what Rosa said, that we need to collaborate. So I think I see more integration between the genomic or geneticists, molecular breeders, physiologists, and agronomists working on this direction. But I think the most important thing for me is the strategic investment into this direction. Unfortunately, politicians operate by 3 or 4 year cycles, and that's simply not enough to solve the problem. COVID was, again, another great example. Would not these vectors and for vaccines, for other vaccines ready, we will never ever deliver it in a year after the pandemic started. So it's just a result of 20 or 30 years of background work. And I think we need to get the stable funding to look at that, because in 20 years, I think we'll have simply no choice and we'll be really dealing with the emergency issue. So we need really to think now, and scientists are normally bad communicators. We cannot talk to politicians. We don't like to speak to journalists or be on TVs, and we take it as granted. Oh, you know, these nerds, they're doing whatever they're doing, and it's not important. It is important, and we need really to think well ahead of that. So we need strategic direction for food security, one or another. I think Michael made it a good point. We really cannot make really decision which way is better. So not to putting all eggs in one basket, we need to pursue both pathways, plus agronomy, of course, which is for third world countries, it's the only solution at the moment, I think. So anyway, but strategic investment is my takeaway message.
Gosh, well done, panelists. That's an absolutely superb kind of summary and last thoughts that kind of cross the whole spectrum there. And yes, I'll put my vote in for, as well as fantastic research, also the communication of that research is super important for all parties really that we need to communicate with. And that's of course something that Frontiers in Science does brilliantly, as we've seen around lead articles like this. It just leaves me then to say a big thank you to today's speakers and expert contributors. Thank you very much, Professor Sergey Shabala, Professor Michael Palmgren, Professor Malcolm Hawkesford, and Dr. Rosa Rivero. I'd also like to thank you for joining us live. I really, really hope that you enjoyed this session. I found it absolutely fascinating. If you can hang on for one more minute, I can tell you about what we have got coming up for you next. To read the article that we've been discussing today, to see all the related content, please visit the Frontiers in Science website. Please do share today's deep dive with your colleagues and continue the conversation on social media. You can use the hashtag Frontiers Forum for that. Be sure to subscribe to the Frontiers in Science updates. That is it from me, our guests, the entire team at Frontiers, who, as always, have done an absolutely incredible job putting all this together. So thank you much to the team behind the scenes. Thank you all again. It's goodbye from all of us. Until next time.
My pleasure.
See ya.
Bye-bye. Thank you.
Thank you all. Bye.